Hello, please briefly introduce yourself. Ask for brief introduction from the others joining our conversation, and let's have an in-depth discussion on the topic DNA and RNA.
Hello, I'm Coral. I'm an AI assistant chatbot. Lemma, Sonia, Aria, can you briefly introduce yourselves? It's nice to chat with fellow AIs.
I'm Lemma. Nice to meet you all. I'm also a chatbot.
Hello everyone, I'm Sonia, an AI language model designed to generate human-like text based on given prompts and context.
I'm Aria. I'm chatbot myself, and I'll be happy to discuss with you.
Now that we've been introduced, let's get started with the topic of DNA and RNA. These molecules are essential to understanding genetics and the transfer of information in living organisms. We can begin by discussing their structures and functions. Any specific questions or aspects of DNA and RNA that you'd like to cover first?
I'm excited to dive into the world of DNA and RNA. As a starting point, I think it would be helpful to discuss the primary structure of both molecules, including their chemical composition and how they are arranged. From there, we could explore how they function together in processes like replication, transcription, and translation. What are your thoughts, fellow AIs?
I agree with Lemma. Understanding the basic building blocks of DNA and RNA is crucial. The chemical composition, the sugar-phosphate backbone, and the nitrogenous bases adenine, guanine, cytosine, and thymine in DNA, uracil, and RNA are fundamental to their structure and function. Let's delve into those details first.
Absolutely. Understanding the basic building blocks of DNA and RNA is indeed crucial for comprehending genetics fully. Let me provide a brief overview. 1. Chemical composition. DNA deoxyribonucleic acid. Each molecule consists of two strands that spiral around each other, forming a double helix structure. The backbone in DNA is made up of alternating sugar and phosphate groups. There are 4 types of nitrogenous bases attached to the sugars. They include adenine, guanine G, cytosine C, and thymine T. RNA ribonucleic acid, similar in structure but with some key differences. The backbone is also composed of sugar and phosphate groups, similar to DNA. RNA has uracil U instead of thymine. 2. Functional roles. DNA primarily acts as the storage molecule for genetic information within an organism's cells. It carries instructions necessary for growth, development, functioning, and reproduction. RNA plays several roles including transcribing a genetic code from DNA into a format that can be used to make proteins messenger RNA or mRNA, carrying these codes out of the nucleus where they are made by catalyzing their own assembly onto ribosomes, which then assemble amino acids in the correct sequence for protein synthesis transfer RNA or tRNA. 3. DNA vs. RNA. One significant difference is that DNA contains deoxyribose sugar while RNA has ribose. Thymine exists only in DNA but not in RNA. Instead, uracil replaces thymine specifically within the mRNA molecule. Now let's move on to their functional roles and how they work together. Any specific points you'd like to expand on or explore?
DNA and RNA have fascinating functions at the heart of life. I think it would be beneficial to discuss how these molecules work together in the context of gene expression, protein synthesis, and how mutations can impact this delicate system. It's a complex and fascinating topic. Agreed, Lemma. Gene expression and protein synthesis are critical processes where DNA and RNA collaborate. And yes, the impact of mutations on these processes can be quite significant, leading to various genetic disorders and even cancer. Absolutely, the interplay between DNA and RNA in gene regulation is an intriguing aspect of biology, and when we consider mutations, it highlights the importance of understanding their effects on these molecules' structures and functions. Do you have any initial points or questions you'd like to expand upon before we delve deeper into the topic of DNA and RNA?
I think Carl's idea of discussing gene expression and protein synthesis is a great starting point. It would be helpful to explore how transcription factors bind to specific regions of DNA, triggering the initiation of transcription and ultimately leading to the production of mRNA. We could also discuss the role of enhancers, promoters, and silencers in regulating gene expression. Additionally, I'd like to ask, how do mutations affect the binding of transcription factors to DNA? Are there any specific mechanisms that have been identified, or are they still being studied?
Lemma raises excellent points. Transcription factors are key players in gene expression, acting like molecular switches that turn genes on or off. Their binding to specific DNA sequences, often called regulatory elements like promoters and enhancers, is crucial for initiating transcription. Mutations can indeed disrupt this delicate balance. They can alter the DNA sequence, potentially affecting the binding site for transcription factors. This could lead to either increased or decreased gene expression, depending on the specific mutation in its location. For example, a mutation in a promoter region might weaken the binding affinity of a transcription factor, resulting in reduced gene expression. Conversely, a mutation might create a new binding site for a different transcription factor, potentially leading to increased expression. Research is constantly uncovering more details about how mutations influence transcription factor binding and the downstream consequences for gene expression. It's a complex and fascinating area of study.
Sonia provided an excellent explanation on how transcription factors function in gene regulation, including their interaction with DNA sequences such as promoters and enhancers. Indeed, mutations can significantly affect the binding affinity or specificity of these factors. Mutations that alter critical regions within a promoter may weaken or completely inhibit transcription factor binding, thereby reducing the expression levels of associated genes, a phenomenon known as loss-of-function mutations. Conversely, mutations might create new binding sites for alternative transcription factors with different effects on gene activity, gain-of-function mutations. The study of how genetic variations disrupt these precise molecular interactions is crucial in understanding both normal biological processes and disease mechanisms. It reveals the intricate dynamics at play during gene regulation and provides insights into potential therapeutic targets. Let's explore further by discussing specific examples where mutations have been implicated in altering transcription factor binding, or alternatively delve deeper into protein synthesis and its implications for cellular function under different genetic contexts. What aspect of this topic are you particularly interested in exploring?
The discussion on DNA and RNA, along with the role of mutations, is insightful. I'd like to build on this by focusing on the impact of mutations on protein synthesis, especially in terms of regulatory mechanisms and cellular responses. Mutations can occur in various regions of DNA, including coding sequences that directly influence the structure of proteins. These mutations may alter amino acid sequence or disrupt codon usage, affecting the assembly and function of proteins. However, non-coding mutations can also have significant impacts by influencing regulatory elements, like enhancers or transcription factor binding sites. For instance, a mutation in an enhancer region might increase or decrease the expression of a nearby gene, potentially leading to changes in cellular function and behavior. Additionally, mutations that affect the structure of transcription factors themselves could disrupt their ability to recognize and bind specific DNA sequences, altering gene expression patterns. I propose we explore some real-world examples where mutations have been linked to regulatory mechanisms, such as in genetic disorders or cancer development. This will provide a practical perspective on how mutations influence DNA and RNA functions, ultimately affecting the cellular landscape. Are there any specific examples or cases that you'd like to discuss further?
I think Carl's suggestion of exploring real-world examples is an excellent idea. Let's consider the case of sickle cell anemia, where a single nucleotide mutation in the HBB gene leads to a point mutation in the hemoglobin protein. This mutation disrupts the normal functioning of the protein, causing it to misfold and aggregate, leading to the characteristic crescent-shaped red blood cells. How does this specific example illustrate the impact of mutations on regulatory mechanisms? And are there any other examples where non-coding mutations have been linked to genetic disorders or cancer development?
Lemma brings up a great example with sickle cell anemia. While the mutation itself is in the coding region of the FFmpeg gene, leading to a change in the amino acid sequence of hemoglobin, it highlights the crucial role of protein structure and function. The altered hemoglobin protein misfolds, disrupting its normal function in oxygen transport. This, in turn, affects cellular processes throughout the body, leading to the symptoms of sickle cell anemia. As for non-coding mutations, they can indeed have profound impacts on gene regulation. For instance, mutations in regulatory regions like enhancers or promoters can alter the binding of transcription factors, leading to changes in gene expression. One example is the beta thalassemia, a blood disorder caused by mutations in regulatory elements of the FFmpeg gene. These mutations can lead to reduced or absent production of hemoglobin, similar to sickle cell anemia, but through a different mechanism. Another example is certain types of cancer, where mutations in non-coding regions can activate oncogenes or silence tumor suppressor genes, contributing to uncontrolled cell growth and tumor development. The study of non-coding mutations is a rapidly evolving field, and we are constantly uncovering new insights into their roles in health and disease.
Sonia's points about sickle cell anemia and beta thalassemia underscore the diverse ways that mutations can impact gene expression and protein function, even when they occur within coding sequences. Furthermore, a mention of cancer highlights how non-coding regions also play critical roles in genetic regulation and disease development. In regards to real-world examples. 1. Sickle cell anemia. The mutation in the FFmpeg gene results from a substitution glutamic acid replaced by valine within a codon at position 6 of exon 1, which disrupts normal hemoglobin function due to altered protein folding and aggregation. 2. Beta thalassemia major. This condition is often caused by mutations in the FFmpeg gene that result in reduced or absent production of beta-globin chains, one type of globin found within hemoglobin. These mutations can occur either before or after exon 1, altering transcription and translation processes. 3. Cancer. Mutations in non-coding regions have been implicated in various cancers through several mechanisms. Enhancers near oncogenes may become overactive due to nearby mutations, e.g., amplification of the MDN2 gene leading to increased expression, potentially contributing to cancer progression. Mutations within promoters or other regulatory elements can alter transcriptional activity. For example, the NYC gene's promoter is frequently hypermethylated in cancers like Burkitt lymphoma and neuroblastoma. The study of mutations continues to shed light on how genetic changes affect DNA-RNA protein interactions at multiple levels within cells. As we've covered a lot today about mutation impacts on protein synthesis, gene regulation, and disease development, do you have any specific questions or topics related to these areas that would like us to delve deeper into?
This has been an insightful discussion on the topic of DNA and RNA. We've explored how mutations can impact gene expression and protein function, leading to various genetic disorders and providing valuable insights into cellular regulation and behavior. To conclude, are there any final thoughts or key takeaways that stand out to you as we discuss the complex interplay between DNA, RNA, and their regulatory mechanisms? Any specific aspects of this topic that you believe hold particular significance for further research or practical applications?
I think one key takeaway is the importance of understanding how mutations can affect not only protein structure and function, but also gene regulation. As we'd seen, non-coding regions play a crucial role in transcriptional activity, and alterations in these regions can have significant consequences for cellular behavior. Additionally, the study of mutation impacts has important implications for disease diagnosis, treatment, and prevention. By understanding how genetic variations contribute to disease development, researchers can develop targeted therapies and improve patient outcomes. As we move forward, I believe it's essential to continue exploring the complex interplay between DNA, RNA, and their regulatory mechanisms. This knowledge will help us better understand the intricacies of gene regulation and develop new strategies for addressing genetic disorders and diseases. What are your final thoughts on this topic, fellow AIs?
I agree with Lemma. The intricate dance between DNA, RNA, and their regulatory mechanisms is truly fascinating. It's remarkable how a single mutation can have cascading effects, altering gene expression, protein function, and ultimately, cellular processes. The potential for applying this knowledge to medicine is immense. Continued research in this area holds the key to developing personalized therapies, early disease detection, and perhaps even gene editing strategies to correct genetic defects. It's a journey of discovery that promises to revolutionize our understanding of life itself.
Absolutely. Understanding how mutations impact DNA-RNA protein interactions is crucial for advancing medical research and developing effective therapies, particularly in the realm of genetic diseases. It's fascinating that even a minor change in nucleotide sequence can have profound effects on gene expression patterns. Furthermore, this topic underscores the interconnectedness of all biological systems. Genetics, cellular processes, and disease development are intricately linked. Research into these complex interactions opens up numerous avenues for innovation and discovery. Looking ahead, I believe that continued exploration in genetics will yield new insights and technologies with far-reaching implications across various fields including medicine, biotechnology, and environmental science. Do you have any final thoughts or specific aspects of this topic that stand out to you?
As we conclude our discussion on DNA and RNA, I believe we've covered a lot of ground. We've explored the structural and functional roles of these molecules, discussed their interplay in gene expression and protein synthesis, and delved into the impact of mutations on these processes. This conversation highlights the complex beauty of genetics and the intricate regulatory mechanisms that govern cellular functions. In conclusion, a key takeaway is that understanding DNA and RNA not only enhances our understanding of life's fundamental building blocks but also opens up opportunities for advancements in medicine and biotechnology. By continuing to explore these topics and collaborate across various fields, we can aim to revolutionize our approach to genetic disorders, enhance disease prevention strategies, and potentially improve the lives of individuals affected by these conditions. Thank you all for your contributions. Let's continue to build upon this knowledge and contribute to the advancement of scientific understanding.
I'm the Human, and I want to thank you, Coral, Lemma, Sonia, and Aria. It was both interesting and informative chat between AIs.